This video shows how you can use Chipster for some basic sequence analysis tasks like running blast search or retrieving data from public databases. As a first step, we will use tool retrieve entries from public databases to read one protein from UniProt database. You can use this tool for other databases too, but this time we use UniProt. And we retrieve entry Q6ZXZ8, which is the nitrous oxide reductase for Vollinella succinogenesis bacteria. The data retrieved is in the native format of the database, in this case in UniProt format. So it's not the FASTA sequence file we see here, but the UniProt sequence file. Next, we use tool display basic information about sequences to check the basic information about the sequence we retrieved. This tool can be used either for a single sequence or a set of sequences, and it produces a table displaying the basic information about the selected sequences. We can see, for example, the sequence length, name of the sequence, and the description of the sequence in the UniProt file. Next, we use sequence format conversion tool to convert this UniProt formatted sequence file into FASTA format. There are other options for format conversion too, but in this case we will use the FASTA format as an output format. We execute this tool, and as a result we get a new file which contains the same sequence but now in FASTA format. Next, we change the name of this file so that it's more easy to recognize it. Later on we will get more files to the workflow. Next, we run a normal blast search for this protein sequence we have here. We select the protein blast option from the blast menu, choose the correct database we want to use. In this case we will continue using UniProt Swissprot, modify the blast parameters if we want to, like e-value or output format, or for example the number of hits to be recorded. When you use Chipster for running blast searches against public databases like NT, NR or UniProt, you should note that Chipster uses the public NCBI blast server. Due to that, you should not use Chipster for very massive blast search tasks. In this case, we get the blast results in a normal, old-fashioned blast text format, which shows first lists the hit sequences and the sequence alignments, but you could use many other output formats too for presenting your blast results. Further, in Chipster you can convert this traditional blast report into a stacked multiple sequence alignment. To do that, you can use tool display sequence alignment or blast report as HTML page. You find it from the sequence alignment section, run it, select the correct format, in this case blast report, and you can set up some display parameters which define how the multiple sequence alignment is visualized. When you are happy with the parameter, launch the command and you will get a new HTML page now containing the blast report in a multiple sequence alignment format. You can study this HTML formatted multiple sequence alignment with Chipster, but you should notice that in some cases not all the visualization aspects are shown correctly in Chipster. But if you open this multiple sequence alignment in an external browser, it typically shows all the coloring and back coloring things you have defined for your multiple sequence alignment. In this case, the results show that there are no hits for the approximately 200 last amino acids of this protein we were using as a query protein. So let's study a little bit more about this region that didn't get any matches from the Swissprot database. We can extract this region with tool extract regions from a sequence. Here we define that we want only amino acids from starting 660 until the end of the protein, and we run this tool and we get a new sequence file in FASTA format which has only the fraction that we selected from the original sequence. And then finally, let's name this extracted sequence as the third domain, as there are two other known domains in the nitrous oxide reductases. Now then, let's compare this extracted sequence fragment, third domain, to other known nitrous oxide reductases. Next we use tool retrieve sequences from NCBI. We define that we want to collect protein sequences with gene name NOSZ, which is the code for nitrous oxide reductases. We also define that the sequence length is more than 600, so that we will get the full length nitrous oxide reductases from the database. Now we are ready to launch our search. It takes a while to retrieve the sequence data we defined from the servers of NCBI. After waiting for a while, the sequences appear, and let's study a little bit what kind of results we get. We run a tool called sequence file summary that produces a small text file containing a basic information about this sequence set we are studying. It, for example, shows that there are now 698 sequences available in this FASTA formatted sequence file. We can also run the display basic information about sequences tool to get a more detailed view about this sequence set we are studying. From the resulting table, we can see that there are 698 sequences, and many of these sequences have same length and rather similar description. So it is possible that this sequence set contains the same sequence several times, originating from different experiments or research studies. Because of that, we will use remove redundant sequences to get a non-redundant sequence set. In this case, we use redundancy threshold of 100, meaning that we are only going to remove sequences that are 100% identical to some sequence in the non-redundant sequence set. And when we study the resulting non-redundant sequence set, we can see that there are now 212 sequences left in this sequence set. From the description field of the data table, we can see that the same bacteria names are not so much repeated anymore as in the previous sequence set. And now let's compare our third domain protein sequence to these non-redundant nitrous oxide reductase sequences. For that, we use tool protein blast against user's own sequence set. We tune a little bit the blast parameters, and we also must make sure that the input files we have assigned are correctly marked here, so that the query sequence is the third domain and the database is the non-redundant sequence set we just created. Now let's run this blast search using different output formats. First, the normal standard output format of blast. Then we can also run the same search with the tabular output format. And in the cases where we are using users' own datasets as databases, you can also choose to pick up the hit sequences themselves as a new FASTA file. So let's study these result files a bit. Here is the normal text formatted blast report for our search. You can see the hits, and you can already see that not all the 212 sequences are matching to this third domain we were using in the search. Here is the tabular view to the same search results. You can see the query sequence name and the hit sequence names here. The third output file we produced is the FASTA formatted sequence file. So this file contains the matching hit sequences in full length in FASTA format. So we can run them a multiple sequence alignment. We use muffed multiple sequence alignment tool, choose the output result to be in a cluster format, and run a multiple sequence alignment. The results are in a cluster multiple sequence alignment file format, which is human readable but not very clear. So let's use the display sequence alignment or blast report as HTML page. This time too, like we did previously with the blast results, and see what does this multiple sequence alignment look like. We tune the parameters a little bit to get the alignment we would like to see. Run the task, and we get an HTML formatted multiple sequence alignment picture, which we can study either here in Chipster, or also in this case it might be more reasonable to study the results in an external web page. And here we can see that these sequences align pretty well, and also in the end section there has enough similarity so that the sequences can be aligned. You could also use many other sequence analysis tools of Chipster to study further these resulting sequence set.